Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing University of Hyderabad MSc Entrance Examination for question paper for the subject Biochemistry conducted in the year 2020, and I will be discussing only the biology questions. So next one, poly-DT deoxythymidyllic acid is from 1 to 25 questions, only one option is correct. Multiple thymine bases, a thymidine nucleoside, a thymidine nucleotide, a nucleic acid. So poly-DT, that is deoxythymidyllic acid, it is a nucleic acid. Answer is option D. Then cytosine on deamination produces uracil. Answer is option A. During photorespiration, the reactive oxygen species H2O2 is produced in peroxisome. Answer is option B. The precursor for the synthesis of heme is glycine. Answer is option C. All these are just direct questions. Then which of the following statement is correct? Trehalose is a disaccharide that contains 2 glucose units. That is correct. Then isomaltose has 2 glucose residues linked in alpha 1 4 linkages. Isomaltose has 2 D-glucopyranose units connected by alpha 1 6 linkages. Then lactose consists of 2 galactose units. Lactose has galactose joined to glucose. Then raffinose is an example of disaccharide. Raffinose, it is an oligosaccharide. It is a trisaccharide made of galactose, glucose and fructose. So only option A is correct. Then in a heterozygous diploid organism carrying 2 alleles for a trait, both the traits are expressed. This is an example of codominant. So answer is option B. The blood group of a universal blood acceptor is AB, RB positive. So answer is option C. Sorry, it is RH positive, not RB. Okay. Then all of the following structures are made of tubulin except lamellipodia, because they are made of actin filaments. So answer is option D. Then which of the following DNA polymerase is used during base excision repair in human cells? So it is pol beta. Answer is option B. Which of the following might be the units of specific activity of an enzyme? So it is micromole per minute per mg. So answer is option B. Which of the following life history strategies is not characteristic of a strictly R strategistic species? Species whose life histories are geared to achieve the maximum possible rate of population growth. So R selected species, they have high growth rate but low survivability. Okay. So we'll see. Generally, rapid embryonic development, rapid maturation to reproductive age and small body size. So that is not correct. Then long lifespan. High density dependent mortality. So not characteristic. Okay. And typical of type 2 survivorship. So that is not characteristic of our selected species. So answer is option B. Then a proglottid is. So proglottid, it is a segment of the tapeworm containing a complete sexual mature reproductive system. Answer is option D. Long filamentous threads protruding at the ends of the young cob of maize are. They are the styles. Answer is option C. Then deficiency of which of the cofactors adversely affect the synthesis of purines and pyrimidines? So it is folate. Answer is option A. Then the sedimentation velocity of a protein in a centrifuge does not depend on. So density of the protein, yes. Mass of the protein, yes. Charge on the protein, does not depend on charge on the protein. Shape of the protein, yes. Then for the reaction where substrate A is converted into product D, the reaction coordinate diagram is given below. The number of intermediates, transition states and the reactants of the rate determining step are dash 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 respectively. Choose the correct combination from the options given below. So the answer here is option A. That is A, 2, 3 and B. So the number of intermediates, transition states and the reactant of the rate determining step are. So rate determining step is 2, 3 and dash respectively B. Choose the correct combination of the options given below. So I have given it in the next. This is the transition state, that is over here, which I have marked. Okay. So this is the transition state. These are the transition state. These B and C, they are the intermediate states. And this one is the rate determining step, because rate determining step in a reaction mechanism is the slowest step. It is characterized by high activation energy. So this is the rate determining step. So the answer is option A. Then next one. Which one of the following unit is a unit of catalytic efficiency of an enzyme? So 70, okay. It is minute per molar. Okay, 19 is option C. Then next one. Aerobic oxidation of glucose or fructose releases 32 ATP molecules. How many ATPs are generated upon complete breakdown of sucrose when metabolized by sucrose synthase? So it is 65 ATP molecules are generated. Then a sodium potassium pump is a type of antiport. Answer is option C. Then which of the following liver enzymes absent from other tissues gives the liver an advantage over other cells in taking up glucose after a meal? So it is glucokinase. Answer is option D. Then given below are the names of infectious diseases causing pathogen and vectors that are linked to the spread. Select the option that represent the correct combination of infectious disease causing organism and their vectors. So Escherichia pestis, it is flea. Answer 1E. Leishmania donovani, Phyllobotomus argentipus. 2B. Borrelia burgdorferi, it is black-legged ticks, and dengue fever, it is Anopheles gambiae. So answer is option C, that is 1E, 2B, 3D, 4A. Sorry, 4C, okay, Aedes aegypti. Dengue is by Aedes aegypti. So the answer here is option B. I made a mistake. Okay. So it is option B, that is the correct answer. The next one. Which of the following statements are true for sponges? So sponges are diploblastic animals. Sponges have specialized organs for digestion, nothing. Then gemmules are internal buds found in sponges and are involved in asexual reproduction. That is incorrect. True in case of sponges. The others are wrong. From 26 onwards till 70, you have more than one correct answer. So you have to mark all the answers which are correct. Only then you will get marks. Okay, then next one. Higher amount of polyribosome fraction in the total ribosome of a given cell might suggest that rate of protein synthesis high. That is correct. There is a block in protein synthesis. Yes, that is also correct. Then the initiation of protein synthesis is high. That is also correct. Then the rate of elongation in protein synthesis is decreased. That is also correct. So higher amount of polyribosome in the total ribosome of a given cell might suggest that rate of protein synthesis high, there is block in protein synthesis and initiation of protein synthesis high, and the rate of elongation in protein synthesis is decreased. Then next one, a bacterial operonic mRNA. So bacterial operonic codes for single peptide has multiple stop codons. That is correct. Answer option B is correct. Has a poly A tail? No, it is for eukaryotes, and has 5 prime phosphate. Answer is option B and D are correct. Then based on the function of a gene can code for a repressor. Yes, you can see in case of lac operon. Then transfer RNA, yes, tRNA, ribosomal RNA, no, and operator, that is also correct. So, sorry, operator it cannot code. Okay, ribosomal RNA it can code. So, answer is option A, B, and C, not D. Then next one. Which of the following condition promote maximal expression of lac operon? So high CAMP, no glucose, no lactose, no use. Then with glucose, no glucose should be absent. In the presence of glucose, glucose has to be absent. High CAMP, lactose without glucose. So that is correct. Option D is correct. Which of the following exist as isoenzymes in humans? So hydratase, no, lactate dehydrogenase, yes, that exists as isoenzymes. Then pyruvate kinase, that is also existing as isoenzyme. Threonine deaminase, no. So answer is option B. Then next one, which of the following has more than one chiral center? So if you take the case of isoleucine, it has got this one. Wait. This one particular carbon atom to which different groups are attached. Then you have got this particular carbon atom to which different groups have been attached. Here no. So two are there in case of isoleucine, and in case of isocitrate also this is one particular carbon atom and this is the second carbon atom to which different groups are attached. So isoleucine and isocitrate or isocitric acid, they are having more than one chiral center. So answer here is option A and B. Then next one. Which of the following biochemical reactions catalyzed by enzyme are reversible? So lactate, threonine deaminase, no. Lactate dehydrogenase, yes. Transaminase, yes. Pyruvate dehydrogenase, no. So B and C reactions catalyzed by the enzyme are reversible. Then glutamine serves as an amide group donor in the formation of glucosamine 6-phosphate, yes, and histidine, yes. A and B. Then urea cycle enzymes. So urea cycle enzymes are found in the mitochondria and cytosol, yes. Facilitate removal of toxic excess ammonia from the blood, yes. Occurs in invertebrates, no. Also found in some plants, no. So the answer is urea cycle enzymes. They are A and B are correct. Then next one. Which of the following statements about digestion and absorption of dietary lipids in humans is correct? Digestion by lingual lipase, gastric lipase, pancreatic lipase, yes. Channeled through lacteal system by chylomicrons, that is also correct. Absorbed by active transport, no. Absorbed by complexing with a tripeptide, no. So A and B are correct. Which of the following traits show polygenic inheritance? Human skin color, yes, it is polygenic inheritance. Sickle cell anemia, no. Phenylketonuria, no. Human height, yes, human height is also polygenic inheritance. So A and D are correct. Then which of the following enzymes can catalyze the formation of phosphodiester bonds? So DNA polymerase 1, yes, it can catalyze. RNA polymerase 2, yes. DNA topoisomerase 2, yes. DNA ligase 1, yes. So A, B, C and D are correct. Then which of the following genes code for enzymes that have RNA dependent DNA polymerase activity? So it is coded by Paul gene of HIV, yes. Then Paul gene of copia retroelement, yes. Paul G gene of human, no. HTERT gene of human, yes. So A, B and D are correct. Then during lagging strand synthesis, to join together two precursor fragments, several enzymatic activities happen in a sequential manner. From the following, pick the relevant activities that are required once the two Okazaki fragments are already synthesized. So 5' 3' polymerase activity of DNA polymerase 3, no, it is polymerase 1. So B is correct. Then ligase activity, yes, that is also required. And 5' 3' exonuclease activity of DNA pol 1, that is also required. So B, C, D are required. Then the B form of DNA, so it is the most common form, yes. It is left-handed, no, it is right-handed. Is stabilized by hydrogen bond between successive residues, not between successive residues between the two strands. Okay, has groups that allow molecules interacting with DNA to recognize base sequences, yes. So A and D are correct. Then catabolic repression, a mechanism of gene control in prokaryotes, is mediated through CAMP, is mediated through CAP, yes, yes. If you see E. coli lac operon, you can see all these things. Results in de novo synthesis of positive activator protein, no. Affects enzymes involved in catabolic reaction, yes. So A, B and D are correct. Lysogenic state of lambda phage is favored when? So lysogenic state is favored when nutrients are limited, yes. Low level of C3, expression of an anti-terminator Q protein, no, no. High CAMP level are found in host cells. That is also correct. So 42 is both A and D are correct. Then next one. Assuming that the IgG and IgE isotypes of antibodies were raised against the same antigenic epitope, which of the following may differ between the two isotypes of antibodies? So order isotypes is the genetic variation or difference in the constant region of the heavy and light chain, that is isotypes. So the constant region of heavy chains, yes. Constant region of light chains, yes. Only the variable region, no, it is the constant region. The J chain can be connected by two monomers, no. So the answer is option A and B. Then next one. Which of the following kinetic plots suggest cooperativity and possible allosterically regulated enzyme? So it is plot A, yes. Reaction velocity, then substrate concentration increases. Okay, so A is correct. Then D is also correct. So these suggest cooperativity and possible allosterically regulated enzymes. So A and D are correct. Then next one. Cell elongation in internodal region of the green plants take place due to gibberellins. Cell elongation in internodal region. So it is gibberellins and diterpenoid acids. So answer is both C and D. Then next one. Which element helps in transport of electrons from water in photosynthesis? So Mn is involved as well as chlorine is also involved in photolysis of water. So Mn, transport of electrons from water in photosynthesis, that is Mn and chlorine are involved. Na and sodium and potassium are not involved. Then next one. The substrate for the enzyme primase is or are DNA, RNA primer, DNTPs. So 47 is options. Sorry. For the primase it is NTPs, not DNTPs. Okay. So answer is option D. D, answer option D, that is NTPs. Then assimilatory powers produced during photosynthesis are. So water and oxygen, no, that is ATP is produced and NADPH2 is also produced. So answer is option C. They are the assimilatory powers. Then which of the following gases is or are used for artificial fruit ripening? So for artificial fruit ripening you use ethyne, yes. A and B, that is ethylene. These are used for artificial fruit ripening. Then which of the following statements are true regarding microtubules? So globular proteins are involved in microtubule lattice formation. Yes. Then microtubules display polarity. Thus, the plus end of the microtubule is terminated by a row of beta tubulin subunit, and the opposite minus end of the microtubule is terminated by a row of alpha tubulin subunit. That is also correct. Then microtubule polymerization involved ATP hydrolysis. Nocodazole inhibits cell division as it inhibits microtubule growth. So D is also correct. So A, B and D. Then what is the characteristic is shared by D-glucose, glucose and D-glucose? They are both uh, non-reducing sugars. Both are pyranose. Yes. The pyranose forms are mirror images to each other. No, they both have two anomeric forms. So answer is B and D are correct. Then which of the following statements are true for enzymes and catalysts? Both affect rate of reaction. Yes. Both decrease the activation energy. Yes. Both increase the rate of reaction at similar scale. No. They are both specific in nature. No. So A and B are correct. Then which of the following evidences suggests that mammalian mitochondria originated from an endosymbiotic proteobacteria? So circular DNA, yes. Presence of a double membrane, yes. 60S ribosome, no. Similar genome size, no. So evidence which suggests that mammalian mitochondria originated from endosymbiotic probacteria are circular DNA and presence of double membrane. So both A and B are correct. Then which of the following cell junctions are involved in anchoring the extracellular matrix to the cell? So gap junctions, no. Focal adhesions, yes. Hemidesmosomes, 4. So not desmosomes. So for focal adhesion and hemidesmosomes are involved. So answer is 2 and 4. Then next one. Identify the correct statement about glucogen synthase and glycogen phosphorylase. Both of the enzymes are active upon phosphorylation. No. Active form of glycogen synthase is dephosphorylated. Yes. Phosphorylated form of glycogen phosphorylase is active. Yes. Both the enzymes are inactive upon phosphorylation. No. So the answer here is option C, that is 2 and 3. Then identify the cellular localization of these two enzymes, ornithine transcarbamylase and arginosuccinate lyase. So OCT, that is ornithine transcarbamylase in cytosol, OTC and ASL in cytosol, no. It is OTC, ornithine transcarbamylase is in mitochondria and ASL is in the cytosol. So answer here is option B, that is 3 only. Then next one. Identify the correct sequence of reactions found in citric acid cycle. Pyruvate to citrate to oxaloacetate, no. Citrate alpha ketoglutarate isocitrate, no. Citrate aconitase isocitrate, yes, that is correct. Succinate fumarate malate, that is also correct. So 3 and 4 are correct. So option C is correct. Then next one. Which of the following is or are the measure of dispersion? So range is a measure of dispersion. Yes. Mean, no. Mean deviation, yes. C and quartile deviation, yes. They are all measures of dispersion. Then which of the following denotes the active form of the enzyme? Dephosphorylated adenylate kinase. No. Dephosphorylated glucogen synthase, yes. D. Phosphorylated glucogen synthase, no. Phosphorylated adenylate kinase, no. So active form of the enzyme is dephosphorylated glycogen synthase. So answer is option B. Then which of the following provide atoms to form histidine? So phosphoribosyl pyrophosphate, yes. Then glutamine, yes, and ATP, yes. Okay, this is part C and only one right answer is present. So first one, you want to clone a gene in a plasmid. The gene is flanked by BAMH1 on one side and KPN1 on the other side. The plasmid has unique EcoR5 site. So there is a gene. Okay, you want to clone the gene in the plasmid, and this particular gene, it is in the plasmid, it is flanked by BAMH1 on one side and KPN1 on the other side. So KPN1 BAMH1 restriction, they produces cohesivant, and the plasmid has unique EcoR5 site. You have noticed the digestion with BAMH1 and KPN1 produce 5' overhangs and 3' overhangs respectively. Yes, and EcoR1 is a blunt end cutter. So now you have to ligate BAMH1 and KPN1 and EcoR5. So both have to be converted into blunt ends. So wait. Your advisor suggests you to convert the overhangs to blunt ends either by fill-in or by resection, and then clone the insert with the newly generated blunt ends into the plasmid that has been digested with EcoR5. So which of the following would be the most appropriate order of this multi-step cloning process? So you have got to you want to clone a gene in a plasmid, and the gene is flanked by BAMH1 and KPN1, and the plasmid is having EcoR5. So you have to ligate this particular gene to the plasmid, EcoR5. The problem here is the gene being flanked with BAMH1 and KPN1, it produces cohesivance on restriction digestion, whereas the plasmid EcoR5, it is a blunt end cutter. So you cannot join this particular cohesivant to a blunt end cutter. So what you have to do is BAMH1 and KPN1, you have to convert them into blunt end sequence and then join with EcoR5. Okay. And one more thing is that when you are using blunt end, you need not require to prepare the insert to be restriction digested. Okay, that's just my knowledge I am sharing. Okay, now whatever. Okay, we will see what happens. So they have given digest the insert with KPN1 and BAMH1, fill in the overhangs with clinO-DNA polymerase DNA, ligate it to with EcoR5 digested plasmid. Then they have given the procedure how you will be using it to clone. So they have one clue they have given is that advisor suggests you to convert the overhangs to blunt ends either by fill-in or by resectioning, and then clone the insert with the newly generated blunt ends into plasmid. So this blunting, it is a process by which single-stranded overhangs created by restriction digest is either filled in by addition of nucleotides on the complementary strand using the overhang as template for polymerization, or by chewing back the overhang using an exonuclease activity. So this blunting procedure I will explain in the next slide how this particular process is used for cloning the gene into the plasmid. So this is the BAMH1 gene sequence, and it produces 5' overhangs. Wait one moment. Okay, so this will cut here, so it will produce cohesivance. So you have got the 5' overhangs over here, and then KPN1, this is the sequence, and it will cut over here near the 3' end, and so it will produce 3' overhangs. And now you know that they have provided you with clino fragment, and it has got polymerase and 3' 5' exonucleate activity. So what will clino do is, so clino is a 3' 5' exonuclease activity degrades the 3' overhangs from KPN1. So this is the KPN1 sequence. These are the 3' overhangs of KPN1, and these are the 3' overhangs of KPN1, and these will be degraded by clino fragment and its activity, that is 3' 5' exonuclease activity. So all these particular, that is GTAC and CATG, will be digested off by clino fragments 3' 5' exonuclease activity, and you will get something like this, which is blunt end. So there are no overhanging sequences now, they have been removed. So this you will get a blunt end region. Then BAM H1 digest. Now it is having that 5' overhangs are there. So what you have to do is you can fill in the overhangs. Now this is the case where I told you that blunting. So blunting is the process by which single strand overhang created by restriction digest is either filled in by the addition of nucleotides on the complementary strand using the overhang as template for polymerization. So here this portion will be used as the template and it will be used to fill the sequence over here. Similarly, this portion will be used as template and it will be used to fill the sequence over here. So it will fill in the overhangs. See, G, G, A, T, C, C, T, A, G. So it has filled and it has resulted in blunt end, blunt ends. Okay, so here you have got one blunt end and here also you have got one blunt end. Then another way of blunting is by chewing back the overhang using exonuclease activity. So clino is having 3' 5' exonuclease activity, and I showed you in case of KPN1 which is having 3' overhangs, this particular region is chewed off and it has created blunt ends. And now you can clone your plasmid. So the answer will be option C, that is digest the insert with KPN1, as in here. Resect the overhang with clino DNA polymerase. So here you have to resect the overhanging end, that is remove the overhanging ends using a clino DNA, that is DNA polymerase, that is 3' 5' exonuclease activity of clino will cause blunt end formation by removing the 3' overhangs. Then digest the DNA with BAM H1. Again you digest the DNA with BAM H1. Fill in the overhang with clino DNA polymerase and DNTPs. So this is the region where you have to fill and it fills the overhangs. That is this clino along with DNTPs, it will fill the overhangs. Then you have to ligate it to the EcoR5 digested plasmid. Finished. Your cloning is done. So answer is option C. Then next one. What would be the expression level of beta-galactosidase in the presence and absence of lactose in E. coli bacterial strain harboring partial diploid of lac operon as shown here? So OC I plus Z plus and O plus I minus Z plus. So here it is OC, that is constitutive operator. Constitutive operator, it will produce beta-galactosidase irrespective whether lactose is present or lactose is absent. So it will keep on synthesizing beta-galactosidase. Now this is the normal operator and there is no repressor. So since the absence of repressor, it will also keep on producing beta-galactosidase. So what is the question here? What would be the expression level of beta-galactosidase in the presence and absence of lactose in E. coli bacterial strain harboring partial diploid of the lac operon? So now when lactose is present, so I minus, so the repressor or repressor protein, it is mutated, and then O plus, so it will allow RNA polymerase to bind to the promoter and beta-galactosidase will be synthesized. Then it is OC here, so it will also synthesize beta-galactosidase. So with the inducer, Okay, that is over here. With inducer, it will be maximum. So with inducer present, it will be maximum, and without inducer, it will be half maximum. Okay, so the answer here is option C. Then next one. In a test cross, the phenotypic ratio of the two traits was found to be 1:1:1:1. Which of the following statements about this cross is incorrect? Okay, the self cross of the F1 from homozygous parent is likely to produce 9:3:3:1 ratio. This is a typical Mendelian cross. So naturally, when you self the F1, you will get a 9:3:3:1 ratio. So that is correct. Then the genotypic ratio is 1:1. You can see over here from this picture, the genotypic ratio is 1:1:1:1. Then the two traits are segregating independently. That is also correct. The two traits are linked. No, they are not linked. So the incorrect statement is option D. Then given below are the kinetic parameters of an enzyme with or with inhibitors X and Y. So without inhibitor, with inhibitor X, with inhibitor Y. So without inhibitor, Km is equal to 3 millimolar and Km is equal to 2.75 millimolar in case of inhibitor X, and inhibitor Y, Km is equal to 3 millimolar. So now if you take option B, that is X is uncompetitive inhibitor while Y is non-competitive inhibitor, we will see that condition. Now X is uncompetitive. So uncompetitive inhibitor binds to the enzyme substrate complex and not to free enzyme. Whereas non-competitive inhibitor, when the inhibitor binds to the enzyme at the location other than the active site, the inhibitor does not prevent the binding of the substrate to the enzyme, but sufficiently changes the shape of the site at which catalytic activity occurs. Then, non-competitive inhibitors bind equally well to the enzyme and enzyme substrate complex, whereas uncompetitive then uncompetitive inhibitor, they reduce the Vmax and reduce Km value. So here you can see that with inhibitor X there is a reduction in Vmax value as well as the Km value from the condition without inhibitor. So with inhibitor X, Km value has reduced as well as Vmax has reduced. So it is the case of an uncompetitive inhibitor. And whereas in the case of non-competitive inhibitor, there is reduced Vmax and Km is the same. So Km has got remained the same whereas Vmax has reduced. So it is an example of non-competitive inhibitor. So the answer is X is uncompetitive inhibitor while Y is non-competitive inhibitor. So option B is correct. Then next one. The pK1 and pK2 of aspartic acid are 1.88 and 9.6 respectively. Its side chain pKa is 3.65. Calculate the pH where aspartic acid will not move in an electric field. That is the isoelectric point we have to calculate. So Pi is equal to pK1 plus this pKa. Okay. This one pKa 3. I have written by mistake as pKa2. Okay, that is 3.65. So 1.88 plus 3.65 divided by 2, that is 2.77. So the answer here is option A, that is 2.77. The next one, match the items in two columns and choose the correct answer from the following options. So endoplasmic reticulum. So endoplasmic reticulum is glycosylation 1, 2. Mitochondria is lipid oxidation 2, 1. And vacuole is hydrolytic enzymes 3, 4. And plasma membrane is 4, Na K pump, that is 4, 3. and peroxisome is lipid oxidation 5 1. So the answer here is option D. The next one, find the mean deviation from the mean from the following data. So this is the data that is given. So first we have to calculate the mean. So add everything, and how many are there? Total 8 are there. Divide by 8, you will get 10. Then these are the values that they have given, and distance from 10. Distance from the mean, that will be 4 minus. So 6 minus 10, that is 4, 3, 0, 2, 3, 6, 2, 10. Now mean of the distances. This particular distances we have to calculate the mean. So add everything together, divide by 8, you will get 3.75. So the answer here is option A, that is 3.75. Then next one. Which of the following statements are true for endoplasmic reticulum? Smooth endoplasmic reticulum is involved in lipid synthesis. Yes, that is correct. It controls secretory protein synthesis. Yes. It regulates protein glycosylation. Yes. It is a control center of protein phosphorylation. No. So A, B and C are correct. So the answer is option B, that is A, B and C. Then 10 microliters of a protein solution is diluted to 990 microliter of a buffer solution. Then 50 microliter of this diluted sample is added to 450 microliter of the buffer. The absorbance of this solution is determined to be 0.1 in a 1 centimeter cuvette. If the extension coefficient of the protein is 1, what is the starting concentration of the protein solution? So first we have to calculate the dilution factors. Okay. So this is the total volume, that is 1000 microliter. How did you get 990 diluted to 10 microliter of protein sample is diluted to 990 microliters. So 990 plus 10, that will be 1000 microliter, and sample volume is 10. So dilution factor is 1000 divided by 10, that is 100. Then next case, total volume is equal to 500 because 450 plus 50, and sample volume is 50 microliter. So the dilution factor is 10. Then Beer-Lambert's law, A is equal to epsilon Cl, and final concentration, that is this particular C, is equal to A by epsilon Cl. So 0.1 divided by 1 into 1, that is 0.1 mg per ml. Now final concentration into dilution factor is equal to initial concentration. So 0.1 into 100 into 10 is equal to initial concentration. Our initial concentration is 100 mg per ml. So the answer here is option A. So next question. When a mixture of protein M, molecular weight 20 kDa and another protein X is run on a native gel, there is retardation of mobility of protein M during gel electrophoresis as compared to pure protein M. So when a mixture, that is M and X, are run together, there is retardation in mobility, whereas when M is run alone, there is no retardation in mobility. So based on the result, which of the following are likely to be correct? So protein M has no interaction with protein X. That cannot be true, because when protein M is run alone, there is no retardation in mobility, whereas when it is run with along with X, there is retardation of mobility. So there is some interaction between M and X. Then protein X degrades protein M. That doesn't happen here. Only retardation of mobility is seen. Then dye labeled with protein M will definitely show a fluorescence resonance energy transfer signal with dye labeled protein X. So that is also not correct. Then dye labeled protein X may show increased fluorescence anisotropy when incubated with protein M. That is correct. So answer is option D. Then next one. In an interrupted mating experiment between donor who is H plus STR R, TONR and LAC plus, the recipient was found to be LAC plus in 5 minutes, histidine plus in 10 minutes, STR in 15 minutes. However, the strain almost never was TONR in the course of the experiment. So based on this data, which one of the following statements about the position of origin is correct? So now they have told that TONR was not transferred to the recipient, so it must be at the other end. Since lag plus was transferred first, in the 5 minutes itself lag plus was transferred first, and tone R was not transferred in the course of the experiment. So because when the two strains of bacteria got separated from each other after STRR was transferred in 15 minutes. So tone R must be at one end and lag plus must be at the other end. So which one of the following statement about the position of origin is correct? So it is between TONR and lag plus. Answer is option D. Then next one. In aerobic organism, glycolysis is followed by citric acid cycle. Match the enzymes listed in column A to column B. So pyruvate to acetyl-CoA, that is mitochondrial matrix. So A2. Phosphoenol to pyruvate that take place in cytosol. B1. Succinate dehydrogenase. It is present in inner mitochondrial membrane. C4. Fumarate to malate in outer mitochondrial membrane. In mitochondrial matrix, that is D2. So A2, B1, C4 and D2. So option B is correct. Then for each of the item listed below, which of them are indicative of a catabolic pathway? So consumption of ATP, reduction of compounds using NADPH, yielding energy. That is correct. Involvement of the cofactor NAD+, that is also correct. Production of glucose by photoautotrophs, no decrease in molecular complexity. F is also correct. So C, D, F are correct. So the answer here is option D. So that's it. Thank you for watching my channel. Please like, share and subscribe. Thank you.